Hello dear students, welcome to the Botany online class. So today in this video we are going to study about the new topics ultrastructure of cyanobacterial cell and ultrastructure of heterocyst. So now first let us take the topic ultrastructure of cyanobacterial cell. So what do you mean by cyanobacteria? Cyanobacteria are commonly called as blue-green algae. So in this video, we are going to study about the ultrastructure of the cell of blue-green algae. Coming to the structure, the cyanobacterial cell contains two main parts. They are outer cellular covering and cytoplasm. So each cell of the cyanobacteria contains two main parts. They are outer cellular covering and cytoplasm. This outer cellular covering, it can be differentiated into three parts. They are slime layer, and this layer is also called as mucilaginous sheath. The second part is cell wall, and the third part is plasma membrane. So once again I will repeat. The cell of the cyanobacteria, it mainly contains two parts, the outer cellular covering and the cytoplasm. The outer cellular covering is differentiated into three regions: slime layer, cell wall and plasma membrane. So this slime layer, it is also called as mucilaginous sheath. The presence of mucilaginous sheath is a characteristic feature of cyanobacteria. And function of the mucilaginous sheath is it retains the absorbed water and protects the cell against desiccation. That means this slimy layer which is present in the cyanobacteria, it protects the cell from dryness. Okay. Now cell wall is present between the slime layer and plasma membrane. So next to the slime layer, there is a second region called cell wall, and this cell wall is present between the slime layer and the plasma membrane. And the main constituent of cell wall is peptidoglycan. So peptidoglycan is a wall material found in the cell wall of the cyanobacteria. Am I clear? Now, the cell wall is made up of four layers. They are L1, L2, L3 and L4. Okay, the cell wall of the cyanobacteria is divided into four layers: L1, L2, L3 and L4, each of which is connected with the other one by a connection called plasmodesmata. That means these four layers are interconnected with each other with the help of a connection called plasmodesmata. Am I clear? Okay. Now L1 it is a transparent space and it occurs between the L2 and plasma membrane. So L1 layer is transparent and it occurs between the L2 layer and plasma membrane. Whereas L2 and L3 are mucopolymer, that means they are made up of alanine, glucosamine, peptidoglycan, muramic acid, glutamic acid and alpha diaminopimelic acid. Okay, is that clear? So L1 layer is transparent and it is present between L2 and plasma membrane, whereas L2 and L3 layer are made up of alanine, glucosamine, peptidoglycan, muramic acid, glutamic acid and alpha diaminopimelic acid. The L4 layer is undulating, means it is wave-like and made up of liposaccharides and proteins. So the fourth layer of the cell wall, that is L4 layer, is wavy in structure and it is made up of liposaccharides and proteins. Okay, next to the cell wall, the third region is plasma membrane, And this plasma membrane is present below the cell wall and it is made up of protein lipid protein layer. It is 70 angstrom thick and generally fuses with the photosynthetic lamellae and gives rise to inward foldings in the cytoplasm called lamellosomes. What do you mean by lamellosomes? The plasma membrane, it fuses with the photosynthetic lamellae and gives rise to inward folding in the cytoplasm. So this plasma membrane, it forms a inward folding in the plasma membrane and this inward folding is called as lamellosomes. Am I clear? So these are about the first part of cyanobacterial cell. Coming to the second part, that is cytoplasm. Now again this cytoplasm also divided into two regions. They are chromoplasm and centroplasm. Chromoplasm, it is the outer or the peripheral pigmented region, consists of flattened vesicles called thylakoids. And these thylakoids are also called as photosynthetic lamellae. Once again I'll repeat, chromoplasm is nothing but it is the outer or the peripheral pigmented region, contains flattened structures called thylakoids or photosynthetic lamellae. And these lamellae contains pigments such as chlorophyll, carotenoids, phycobilins, phycocyanin, allophycocyanin and phycoerythrin. And the second part is centroplasm. So centroplasm is the inner or the central colorless region, whereas chromoplasm is the colored portion and centroplasm is the colorless region, and it is often called nucleoid or incipient nucleus. And this region consists of DNA fibrils, and here DNA is not surrounded with protein materials. That means histones are entirely absent. Like bacteria, small circular DNA segments occur in addition to the nucleoid, and these are known as plasmids or transposons. So in centroplasm, small circular DNA segments are produced in addition to the nucleoid, and these small circular DNA are known as plasmids or transposons. So these are about the centroplasm. So this is the ultrastructure of cyanobacterial cell. So in this picture you can see the lamellosomes, photosynthetic lamellae, the sheath means mucilaginous sheath, the cell wall and the plasma membrane, and inside the cytoplasm it contains DNA, ribosomes and the granules, etc. So coming to the second topic, ultrastructure of heterocyst. These heterocysts are the large thick-walled cells found in the nostoc filaments. They are pale yellow in color and globose or ovoid in shape. They may be terminal or intercalary in position. Terminal means they are present at the tips, or intercalary means held in between the cells. And the function of the heterocyst is they fix atmospheric nitrogen. A heterocyst develops from the ordinary vegetative cell. The heterocyst, it has a thick wall which is differentiated into two layers. The outer layer is called as outer wall and the inner layer is called as inner wall. So each heterocyst, it contains a thick wall which is differentiated into outer wall and inner wall. And the cell wall of the heterocyst is made up of polysaccharides. And these two wall layers, that means the outer wall and the inner wall, are separated by an empty space. That means there is a space between the two layers, between outer and inner layer, and this space is called as empty space. The wall leaves a pore at each end, and this pore is called as polar pore. That means in the heterocyst, at both the ends, the wall of the heterocyst contains a pore, and this pore is called as polar pore. In mature heterocyst, the pores are closed by a button-like plug called polar nodule. So in mature heterocyst, the pores are closed with the help of polar nodule. And this polar nodule is composed of solid mucilaginous substance. Inner to the cell wall, there is a plasma membrane that surrounds the protoplasm. And in the protoplasm, the cell contains, the heterocyst contains thylakoids and ribosomes and are fewer in number. Along with the thylakoids and ribosomes, globules of glycolipid and acrylipid are also seen in the protoplasm. So this is the structure of heterocyst, cell structure of heterocyst. And in this picture you can clearly observe the two wall layers. They are inner wall and outer wall. In between the wall layers there is an empty space, and at the polar regions polar pores are present, and these pores are they are closed by the polar nodules. And next to the wall layer there is a cytoplasm, and in the cytoplasm it contains the cell organelles like ribosomes, and along with the ribosomes thylakoids are present, and some globules are also present. And in this picture you can see the terminal and the intercalary heterocyst. Terminal means if the heterocyst is present at the tip, then it is called as terminal, and if the heterocysts are present or held between the cells, then it is called as intercalary. So these are about the ultrastructure of heterocyst. Thank you.